Dr. Sarah Ellis is a naturopathic physician and a clinical science liaison with Microbiome Labs. Dr. Sarah has spent the last 10 years investigating the safety and efficacy of natural products. As a naturopathic physician, she uses nutrition, nutraceutical supplementation, and lifestyle adjustments to help her patients get to the root cause of their health concerns. She specializes in women's health, gut health, nutrition, and family health. Okay, Sarah, take it away.
All right. Thank you so much for that awesome introduction. Hi, everyone. I hope you can hear me well, and I hope you can see my slides. Hi, Shelly. I can understand the chat can be a bit distracting. You may be able to close the screen. But either way, I'm so excited to be in front of you all today. We are going to be chatting about the connection between the gut microbiome and our hormones. Healthy gut, healthy hormones. There we go. All right, so today we are going to be covering a brief overview of the endocrine system. I'm going to highlight some common causes of hormonal imbalance, and we'll discuss the impact that gut dysbiosis has on endocrine function. And then the biggest time that we'll spend together, we'll be discussing the specific ways in which the gut microbiome impacts the HPA axis, thyroid function, glucose regulation, and reproductive hormone regulation. And as a new feature, we are going to include a case study at the end. So take some notes, think a little bit about what products you would use once we get to the case, and then I would love some interactive feedback in the chat once we get to that part. So let's get right in and get started. So I'm sure you all know that the endocrine system is a complex network of glands and organs that secrete hormones that help control a variety of activities in our body. They include growth and development, metabolism, sexual function, reproduction, and mood. Now, we have a number of glands, endocrine glands in our body. The major ones include the pituitary gland and pineal glands in the brain, thymus and thyroid, pancreas, the adrenals, testes, and ovaries. Oh, and also I left out the main one of our biggest endocrine glands, which is the gut microbiome. So let's talk about some common causes of endocrine disruption. And we'll start with some symptoms of hormonal imbalance. So common symptoms that your patient may have that are rooted in hormonal imbalances include stress, acne, changes in hair, either thinning hair or hair loss, changes in bowel habits, constipation and diarrhea, difficulty sleeping, excessive facial and body hair, depression, headaches, and much more. All of these symptoms are tied to items, foods, and situations that lead to endocrine disruption. So here are some common causes that you may see with some of your patients. A genetic predisposition. Of course, if someone has a family history of type 2 diabetes or thyroid autoimmune conditions, that is going to be a common cause of endocrine disruption. Other causes include infection, injury to the endocrine gland. It's common that some patients who have a traumatic brain injury may also show up or pop up with endocrine disorders due to the potential damage to the endocrine glands in the brain. Tumors can cause endocrine disruption, along with stress, toxin exposures, liver dysfunction, a poor diet, along with obesity and gut dysbiosis. Now let's take a moment to discuss the gut microbiome and what a healthy microbiome looks like in comparison to a leaky gut. And so here on the left, I'm sure you've seen the schematic a number of times, but here on the left, we can see that a healthy gut has a number of different characteristics. It has increased microbial diversity with a plethora of good microbiota. There are low levels of pathogens. There's a nice and thick and strong mucosal barrier that protects the enterocytes from having direct contact with the bacteria. There's low mucosal inflammation, strong tight junctions between these enterocytes. And there's also an increased production in postbiotics like short chain fatty acids, like butyrate, acetate, and propionate. Now, this is important because short chain fatty acids that are produced by healthy bacteria in the gut directly activate receptors on the cell surfaces of our colonocytes, and they influence the production of hormones like glucagon-like peptide 1 or GLP-1, ghrelin, and leptin. Now, on the left here, or excuse me, on the right here, let me put on my laser pointer. On the right here, we see a leaky gut that has quite a bit of pathogens that are overabundant, and they degrade the mucus layer and produce toxins. They also activate the immune system and our inflammatory cascades. Now, it's important that I highlight that because now we're going to talk about the different risk factors that put your patient at higher risk of developing gut dysbiosis. Infection, excessive alcohol, smoking, stress, certain medications, including natural antimicrobials, poor sleep, intense exercise, a poor diet, and toxins. Now, you may notice that these dysbiosis risk factors are some of the same causes of endocrine disruption. So there's definitely a connection between the gut microbiome and the development of endocrine disorders. Now, I mentioned earlier that the gut microbiome is our biggest endocrine organ. The GI tract is the largest endocrine organ in mammals and is known to secrete dozens of hormones, peptides, and other signaling molecules. Now, when the composition of the gut microbiome is changed, whether negatively by smoking or intense exercise, or positively with healthy fiber, like fiber from kiwi, it changes the gut microbiome and changes who is in the gut. And as we change who is in the gut, it's going to change the postbiotics and metabolites that they produce. Some of the metabolites that they produce can cross the intestinal cells and translocate into circulation and can impact autoimmune conditions and the immune system activation, as we've seen in a lot of autoimmune conditions like type 1 diabetes or type 1 diabetes here and Hashimoto's thyroiditis with the thyroid. Now, what we know is that when you have a healthy gut, you have healthy hormones. All right, so our microbiota makes a ton of different products. They make short chain fatty acids, which we just talked about, choline metabolites that go on to help produce a neurotransmitter called acetylcholine, tryptophan, which is an essential amino acid that our body uses to produce serotonin and melatonin, and neurotransmitters like GABA, which are often produced by our gut microbiome. By focusing on harnessing a healthy and creating a healthy gut microbiome, we're going to be able to have microbiota that are able to make healthy hormones and neurotransmitters and keep our endocrine system in balance. What's also equally important is knowing which bacterial strains make which neurotransmitters. I've included a list that isn't necessarily exhaustive, but I listed some species of bacteria that we're all familiar with, and especially those that we find and can find on our BiomeFX stool test. So let's look at the bacteria that make serotonin. There are a number of lactobacillus species that make serotonin, but there are also some pathogens that do as well, including E. coli, especially the rather toxic strain, the K12 strain, Morganella morgani, Klebsiella, and pneumoniae. So when you see these pathogens pop up on a patient's BiomeFX, you really want to take a look at their chief complaints. Are they presenting with complaints that are associated with low serotonin, or maybe serotonin that's too high? The same can be said for dopamine. There are a series of bacteria that make dopamine, In pink, I highlighted Bacillus subtilis. This is one of our bacillus spores that's found in 4 of our spore probiotic products: Megaspore Biotic, Restore Flora, Serene Skin, and as well as HU58, which only contains Bacillus subtilis. Much like serotonin, there are a number of pathogens that also produce dopamine. E. coli, both the regular basic species and strain, along with the K12 strain, Morganella morgani, Klebsiella, and Hafnia as well. There are also a few bacteria that make noradrenaline, including Bacillus subtilis. Lactobacillus plantarum is known to create acetylcholine. And then there are a number of species known to create GABA. Primarily, Lactobacillus and Bifido species are known to create GABA. These species in the blue, Bifidobacterium adolescentis and Lactobacillus rhamnosus, these are 2 keystone species that we measure on BiomeFX. And so if you see these species present in your patient's microbiome, then you at least know that they have the microbial machinery to make GABA. I also highlighted here Bifidobacterium longum 35624. This is one of the 2 strains present in our ZenBiome Dual product, and it is known to produce GABA as well. The last one that I wanted to touch on was histamine. You may be surprised that there are a number of Lactobacillus species that make histamine, including Lactobacillus plantarum. Now, this is important to note because I'm sure as functional medicine practitioners, you are definitely having your patient on a probiotic, and a lot of the traditional probiotics on the market have a wide array and mix of Lactobacillus species. So it's important to look at the ingredient label and see if any of these species are present in that probiotic. And especially if you're working with a patient that is histamine sensitive or has mast cell activation syndrome, you may want to avoid some of these Lactobacillus species until you're able to regulate the gut and reduce their histamine sensitivity. So now that we've had a solid foundation, we've gotten an overview of the endocrine system, and we've gotten a sneak peek on how the bacteria impact our gut microbiome and our endocrine system, let's get into some specific details. First, we're going to talk about the microbiome and the HPA axis. Our buddy here looks pretty stressed, and as we know, that's not good for his brain or his gut. Now, not only does the brain affect the gut, but the gut also affects the brain. And this is due to a bidirectional influence of the enteric nervous system that powers the gut and the vagus nerve. It's our longest nerve that starts at the base of our skull and goes all the way down and innervates the gut. Now, this bidirectional influence does create a sort of cyclical problem, because if we are chronically stressed, if we have patients that are chronically stressed, they have a poor diet, they're not getting good sleep, and they lead a sedentary lifestyle, that's going to impact their microbiome. And then that change in the microbiome is also going to change the ways in which that patient perceives stress. It changes the type of foods that they crave and their mood as well. Now, there are a variety of host factors, diet, exercise, health state, and mood, that can lead to alterations in hormones and neurotransmitters that are produced by the gut. And in turn, that change in the type and quantity of neurotransmitters does have a significant effect on the microbiome. Here we can see that there are a variety of hormones and neurotransmitters that impact bacterial growth, biofilm growth, decreased resistance to the host, and changes in their virulence. So let's take a moment to talk about stress. We know that stress is considered to be an activator of inflammation. And certain stressors, whether it's early adversity, interpersonal conflict, or even social isolation, can impact the way in which our brain signals our adrenals to release cortisol. When cortisol is released, It can tamp down on inflammation, specifically the cytokines that are activated by NF-kappa B, an inflammatory transcription factor. But this is under an acute situation. And if you're undergoing acute stress, whether it's someone cut you off in traffic, or perhaps you're having an argument with a family member or a friend, that is an acute situation that is easily rectified once the stressor has dissipated. But when it comes to chronic cortisol expression, we see a different picture. So with increased levels of cortisol, we also see an increase in the permeability of the gut wall. So cortisol essentially drives leaky gut. That facilitates a systemic immune response, and that leads to increased levels of circulating pro-inflammatory cytokines like TNF alpha, interferon gamma, and IL-6. Now, I want to take a moment to really focus on interleukin 6. Interleukin 6 is a pro-inflammatory cytokine that's known to activate the HPA axis, the hypothalamus, pituitary, and adrenal axis. Over time, interleukin 6 has been shown to reduce glucocorticoid receptors. In an acute state, that is a way of the body to sort of tamp down on this stress response and cool off the inflammation. However, if you have a patient that has chronic cortisol expression, they're going to have chronic interleukin expression, and then they are chronically going to have low glucocorticoid receptors. Unfortunately, the reduction in glucocorticoid receptors means that they have more free-flowing and free cortisol circulating. There are fewer receptors to bind up that cortisol, and thus it leads to a hyperactive and sensitive HPA axis. These same changes with pro-inflammatory cytokines have also been shown to change the amount of serotonin produced directly in the brain, in the hippocampus. At the same time, they've also seen a reduction in a peptide called brain-derived neurotropic factor. And this is a peptide that has been shown that when reduced, it's associated with depression and mood changes, all of which are associated with dysregulation with the gut and the brain axis. Now, we could talk about cortisol all day, but I really would like to talk about tryptophan and the way in which inflammation and the gut microbiome impacts how our body and our brain specifically is able to use tryptophan. Now, tryptophan is an essential amino acid, and it is used by our body and specifically our brain to create serotonin, which helps stabilize our mood, and melatonin, which helps us have good sleep. Now, the brain does not store tryptophan, so the health of our gut really determines how much tryptophan is available for our brain. In a healthy patient with a healthy gut and limited inflammation, the tryptophan that we get from our diet is able to go into this neuroprotective pathway where it can make serotonin and melatonin. Patients that have this active neuroprotective pathway tend to have better sleep, a more balanced circadian rhythm, a positive outlook and resiliency to stress, as well as a good memory. Now, if you have a patient that has a dysbiotic gut, say they're eating a standard American diet, they have a dysbiotic microbiome, lots of inflammation and inflammatory cytokines, likely a lot of endotoxins like lipopolysaccharide, and unfortunately, if they're also experiencing stress, then that's going to take tryptophan that we get from our diet and drive it into this neurotoxic pathway that leads to the production of quinolinic acid. Now, this is important because, like I said, tryptophan is not stored in the brain. And so all the tryptophan that's going into this neurotoxic pathway, there isn't likely much left to go to the serotonin and melatonin pathway. So if you have a patient that is shuttling their tryptophan into a neurotoxic pathway, they will likely be presenting with a low mood, depressed mood. irritability, low resilience to stress, poor sleep, and poor concentration. All right. So now that we have a good sense of how the gut microbiome impacts our HPA axis, let's take a moment to dive into its effects on thyroid. Now, I have to tell you that this was probably my favorite section of the talk to create because I learned so much about how the gut microbiome impacts our thyroid. So let's get a basic of the thyroid gland. The gut is a target organ for our thyroid. In fact, thyroid hormones, thyroxine or T4, or triiodothyronine, T3, they impact our heart rate, breathing, glucose metabolism, menstrual cycle, liver function, bone and musculoskeletal health, body temperature, body weight, and even brain development. What's interesting is that thyroid hormone receptors are expressed on the surface of intestinal epithelial cells. And in fact, as intestinal epithelial development is controlled by thyroid hormone signaling. And so a disruption in the microbial composition towards a more dysbiotic gut, it has been associated with Hashimoto's thyroiditis, Graves' disease, and Graves' orbitopathy. Let's connect the dots. So in the thyroid, the major thyroid hormone that's produced is T4, and then the minor form is T3. T4 is considered to be the active form or inactive form of thyroid hormone, and T3 is the active one. And so our body relies on enzymes to activate T4 into T3. And of course, the gut makes a ton of enzymes, specifically beta-glucuronidase, which is produced by a group of bacteria called the estrobolome. The gut bacteria can also make sulfatases as well. And so the production of these different enzymes can reverse critical detoxification efforts performed by the liver, and that results in recycling of these two hormones. And as a practitioner, I'm sure you can imagine that this makes it difficult for you as a practitioner to help your patient have stable thyroid levels and kind of get rid of those symptoms that are associated with hypothyroidism or hyperthyroidism. So let's take a look at another connection between the gut and the thyroid. I thought this was really fascinating. Another enzyme that is produced by the gut is called intestinal alkaline phosphatase. This is a brush border enzyme that's produced in the small intestine, specifically in the duodenum. And it works to detoxify LPS. So it deactivates the endotoxin lipopolysaccharide, which is honestly one of the main causes of inflammation coming from the gut. And so this enzyme is able to detoxify LPS so it doesn't increase our inflammation. Now, what's interesting is that the gene that codes for this enzyme is T3 responsive. The levels of thyroid hormone determines whether or not this gene is going to be expressed so the body can make intestinal alkaline phosphatase. And so you can imagine that if you have a patient that has low thyroid hormone, so they're hypothyroid, they're likely not making a lot of intestinal alkaline phosphatase, which means that their inherent ability to detoxify LPS is blunted. Animal models have discovered that hypothyroid rats actually showed a decrease in intestinal alkaline phosphatase gene transcription. And in another animal model with zebrafish, they knocked out the intestinal alkaline phosphatase gene and saw that those animals with the knockout were more susceptible to LPS toxicity. Now, of course, we're going to need some more human studies to really get an idea of the impact of intestinal alkaline phosphatase on human health as it pertains to thyroid disease. But this is a huge reason to clear the gut of patients with thyroid disease so that you can ensure that they're having a healthy thyroid balance. Now, of course, we have to talk about microbial patterns. So here are bacterial strains that are associated with decreased expression of the two primary antibodies involved in Hashimoto thyroiditis and Graves' disease. So the presence of Bacteroides species, the Dorea species, and Faecalibacterium are all associated with a decreased expression of those antibodies. Now, this is important because Faecalibacterium prosnitzi is a keystone. It's a keystone species that it has shown to have a unique impact on the microbiome. And so Faecalibacterium is really important for having a healthy gut microbiome. Now, this is a list of species that have been associated with an increased expression of these antibodies. Now, researchers believe that this is due to molecular mimicry. So the surfaces and proteins on the surfaces of these bacteria mimic these same proteins and receptors involved in thyroid autoimmune disease. These strains include the Blauschia species, Lactobacillus species, Ellis and Ruminococcus species. And then here are 3 species of bacteria that have been seen in patients with Hashimoto's. Of course, this list is not exhaustive, but it highlights 2 pretty common pathogens that we tend to see on our BiomeFX stool test. We have Lachnoclostridium, Bilophila-Wadsworthia, which is a sulfate-reducing bacterium that exists in the bile and makes hydrogen sulfide gas, which can be toxic to the gut. And then, of course, we have Klebsiella, which is, as if you remember in a couple of slides previously, it's one of those that makes histamine, and it makes a ton of other neurotransmitters as well. But it is a pathogen and has been associated with Hashimoto's thyroiditis. Okay. So the gut microbiome and glucose regulation. I'm going to pop in the chat really quick. I see some questions. If you all can pop those in the Q&A, we will answer them at the end. Thank you. All right, so we've covered the microbiome and the HPA axis. We've also covered the microbiome and the thyroid. But now let's look at the gut microbiome and glucose regulation. Okay, so let's talk a bit about these major glucose balancing hormones that are present in the GI tract. Let's take a moment to chat about GLP-1. GLP-1 or glucagon-like peptide 1. If I didn't get that right, someone let me know in the chat. But it's an incretin that has the ability to decrease blood sugar. And it does so by increasing the secretion of insulin. Now, GLP-1 activity is preserved in patients with type 2 diabetes, but it can dissipate over time. And so GLP-1 ensures that beta cell insulin stores are replenished, and that prevents insulin exhaustion. And so it helps create a stable insulin secretion and avoids the development of insulin resistance. One really cool thing about GLP-1 is that it promotes the proliferation and neogenesis of beta cells. So not only does it help us manage our glucose levels by stimulating insulin response and insulin secretion, and also really increases the amount of beta cells that we have in our pancreas so that we can continue to make insulin and support a balanced blood glucose. Next, let's talk about leptin. Leptin is a hormone that's released from our fat cells, and it signals to the hypothalamus that we are full, and it helps stimulate the digestive process as well. Now, it actually alters our food intake and controls our energy expenditure over the long term. And what we found is that obese people have unusually high levels of leptin. And that's because, for whatever reason, likely due to the inflammation associated with obesity, our brain just does not respond to leptin. And so obese patients are much likely to develop something called leptin resistance. Next, we'll talk about leptin's counterpart, ghrelin. This is a hormone that's released by mainly the stomach to let us know that we're hungry. But there are small amounts that's produced in the pancreas, the small intestine, and the brain. It has a number of functions, but of course, we've termed it our hunger hormone. And so it stimulates appetite, food intake, and promotes fat storage. In addition to increasing appetite, ghrelin also mediates whole body glucose homeostasis. Now, in the pancreas, ghrelin stimulates the release of glucagon, which increases the production and release of glucose from the liver. This is usually released in a starvation mode when the brain believes that we are in starvation mode, but can also happen when we are, if you have a patient who is insulin resistant. And then lastly, let's talk about insulin. Insulin is produced by our beta cells in the pancreas, and it is critical for optimal glucose balance. Without insulin, our cells are not able to bring glucose into the cell so that we can make ATP and energy. Now, insulin does have a number of other effects. It can also help with the digestion of our food. It supports the breakdown of fat and protein as well. So what does the research say about gut dysbiosis and type 2 diabetics? Well, we know from a lot of research studies that type 2 diabetics are more likely to have gut dysbiosis. Now, our gut microbiome, it doesn't only impact our leptin levels, our ghrelin levels, but also insulin release and sensitivity. Research has found that Lactobacillus fermentum, Lactobacillus plantarum, Lactobacillus casei, Roseburia, Akkermansia, and B. fragilis have all been shown to improve glucose metabolism, and it suppresses those pro-inflammatory cytokines. Now, what's interesting is that metformin, which is our first-line pharmacotherapy for type 2 diabetes, actually acts primarily in the gut, and it alters the composition of the gut microbiome. Metformin increases the production of butyrate. It increases the production of propionate, both of which of those are short-chain fatty acids. And it also increases the level of a special keystone bacteria called Akkermansia mucinophila. Now, as I said earlier, metformin does act on the gut and alters the microbial composition. This study found that when they looked at the microbial composition, and we look at taxonomy, right? So the broadest group is kingdom, and you can go as narrow as strain. So when we look at the phylum level, researchers were able to see that firmicutes, which is one of the bigger groups of bacteria in our gut, we found that type 2 diabetics actually had lower amounts of firmicutes in their gut microbiome. And this is important because firmicutes do have a lot of keystones. There are a lot of keystone species that exist in that firmicutes group. And so if you're going to have a patient that has fewer firmicutes, they tend to also have fewer keystones as well. The type 2 diabetic patients were also seen to have elevated amounts of proteobacteria. Now, proteobacteria is a very inflammatory group of bacteria that are gram-negative, and they have an endotoxin on their surfaces called lipopolysaccharide, or LPS. Remember, we talked about that a little bit during the thyroid section. Well, if you have a patient that has a lot of proteobacteria, they're also going to have a lot of LPS and thus quite a bit of inflammation as well. Now, what's interesting is that Roseburia, which is a species of bacteria that makes butyrate. Butyrate is a very important short chain fatty acid for our gut. That the abundance of Roseburia was also reduced in those type 2 diabetics. So far, they have low firmicutes, which means they likely also have low keystones. They have a ton of proteobacteria, which means they have a lot of endotoxin and inflammation. And they also have low levels of an important keystone that help makes butyrate. Now, the abundance of these pro-inflammatory strains of bacteria means that they're going to have more inflammation and they're going to have a more stimulated and active immune system. And also, type 2 diabetic patients often had low alpha diversity. So in working with diabetic patients, it's incredibly important to heal their gut and restore their gut so that you have a gut microbiome that can help support healthy blood sugar regulation. All right, so we are at the last section before we get to our case study. Let's talk a bit about how the microbiome plays a role in reproductive hormones. So let's walk through the path of estrogen. Now, in a healthy microbiome, our ovaries are going to make estrogen. Estrogen circles throughout our body, activates a series of receptors. And once it's done doing its thing in the body, it goes to the liver for excretion. The liver will tag estrogen with a sort of chaperone that's going to guide estrogen through the gut and into our stool. And a healthy microbiome with low pathogens and low inflammation, most of that estrogen is going to be pooped out. And then there's going to be a little bit that's going to go back into the body to help maintain estrogen balance. But if you have a patient with an unhealthy microbiome, then their ovaries are going to make estrogen. It's going to circulate throughout the body and activate those receptors, and it's going to go to the liver so that it can be excreted. The liver is still going to tag it with that chaperone, glucuronic acid, and then glucuronic acid escorts estrogen through the gut. But if this patient has an unhealthy gut, so they have pathogens and they have a ton of inflammation, they're likely going to make this enzyme called beta-glucuronidase. Beta-glucuronidase frees estrogen from its chaperone and allows most of the estrogen to go back in the body to circulate again throughout the body and activate estrogen receptors. And unfortunately, only some of the estrogen ends up being pooped out. Now, this process is facilitated by a number of species of bacteria. So when we look at the estrobolome, we are looking at a group of bacteria that makes the enzyme beta-glucuronidase. So let's walk through this schematic again. Of course, we have our estrogens, E1 and E2. It is in the liver where the liver tags it with glucuronic acid, this green molecule here. And now it goes into the gut. But these pathogens are here, like Clostridium, Proteobacteria, E. coli, Salmonella, Pseudomonas, Bacteroides, Shigella, and Streptococcus. They are going to cleave this green molecule and leave estrogen and estrone free, where they can go back into circulation and create an estrogen dominant effect. And so in women, this can present as endometriosis, ovarian cancer, polycystic ovary syndrome, breast cancer, endometrial cancer, and endometrial hyperplasia. Let's take a moment to talk about endometriosis. Endometriosis is an inflammatory estrogen-dependent disease, and it's defined by the presence of endometrial tissue outside of the uterine cavity. It is thought to affect approximately 6 to 10% of reproductive aged women and is a major cause of mortality and morbidity for young women. This research study here looked at the difference in diversity in the control group here in the blue and the endometriosis group in the red. And what they saw is that there was a significantly higher alpha and beta diversity in the control group compared to the endometriosis group. So that means that these healthy volunteers had more bacteria in their gut, and they had a higher quality of bacteria that were health promoting and anti-inflammatory. Whereas those in the endometriosis group had fewer bacteria, and the bacteria that were present were quite inflammatory and pathogenic. In fact, in endometrial lesions, researchers have found a variety of bacterial strains in those endometrial lesions, but particularly they found Fusobacterium nucleatum. Fusobacterium is a species and a group of bacteria that are incredibly pathogenic. They are associated with inflammatory bowel disease and colorectal cancer. And of course, like I said, it's been found in endometrial lesions as well. So if we can remember like the second or third slide that I talked about, we talked about those common causes of endocrine disruption. And one of those on the list were toxins or endocrine disruptors. And they can create a very dysbiotic gut. And so those endocrine disruptors include dioxins, which is a type of pesticide, phthalates that can be found, unfortunately, in everything, plastic bags, personal care products, flame retardants, PCBs or polychlorinated biphenyls, and a host of other endocrine disruptors. They have been associated with dysregulation of intestinal permeability, altered production of neurotransmitters, and chronic low-grade inflammation. Researchers have drawn a correlation between infertility-related conditions and gut dysbiosis. In men, gut dysbiosis can manifest as altered androgen metabolism, a compromised blood-testes barrier integrity, and impaired sperm quality and sperm quantity as well. In women, these gut dysbiosis has been shown to lead to altered estrogen metabolism, premature ovarian failure, polycystic ovary syndrome, and endometriosis. Now, gut dysbiosis can also impact the fetus. There are also a number of other factors that can affect the offspring as well during pregnancy and in the postnatal period. These include the placental environment, which was once thought to be sterile, and now researchers have found that there are bacteria and fragments of bacteria that help the baby develop its microbiome, and it comes from mom's gut. The mode of delivery, breastfeeding, and antibiotics also affect the fetus, along with parental genetics, parental lifestyle, parental microbiota, environment, and maternal stress. Now, of course, we can't have the fertility discussion and just talk about women. Dysbiosis has also been an overlooked factor in male infertility. This study highlighted that E. coli could play a significant role in male infertility. And that's because it's one of the most common infections in men. And also, we've seen that E. coli infection can impact sperm motility, sperm vitality, and sperm morphology. And that's why it was so important to mention the gut dysbiosis and how it impacts the blood and testes barrier. Because if the blood testes barrier is compromised or leaky, that's going to allow bacteria to cross into the testes and impact spermatogenesis and sperm maturation. E. coli has been shown to be able to adhere to sperm and express something called hemolysin A, which is a virulence factor that's been involved in premature acrosomal activation. That's a process that happens as the sperm is maturing and getting ready to travel to meet the egg in preparation for fertilization, but it also can damage that hemolysin A from E. coli can damage sperm nuclear DNA. And we know that if the egg or the sperm have DNA damages, that's going to lead to miscarriages and failed implantation. All right. Shoo, we've made it through the deck. Now it's time to hop in the chat and let's talk about our case study. So, how do we optimize the microbiome to support the endocrine system? All right, so here's our patient. We have a 40-year-old female who is presenting to clinic with a history of hypothyroidism, IBS, and depression. Now, you've made great progress with this patient. She has made some resolutions with her symptoms, but you would love to see a bit more movement and improvement in her symptoms. So, like the forward-thinking practitioner that you are, you ordered a Biome stool test to get a better understanding of her gut health. So let's see what this report came back with. And then in the chat, I want you to tell me kind of what products you would use to address these. All right, so her Biome report shows low diversity. She has low alpha diversity and low beta diversity. So that means she has very few bacteria in her microbiome, and the ones that are there aren't really doing her much favors. She has a high firmicutes to bacteroidetes ratio. Remember, I said that firmicutes is one of the bigger groups of bacteria in the microbiome, and that is second only to bacteroidetes. Ideally, we would love to see a 1-to-1 ratio between firmicutes and bacteroidetes, and in most cases, a slightly less than 1-to-1 ratio in favor of bacteroidetes. But this patient has entirely too much firmicutes and not enough bacteroidetes. And this is a clear sign of dysbiosis. She also has a lot of pathogens. She has C. diff, E. coli, Klebsiella pneumoniae, and Bacteroides fragilis. Now, based on some of the content we've covered today, we know that E. coli can cause inflammation. Klebsiella belongs to the estrobolome, so it's going to affect her estrogen metabolism. It also makes histamine, so it could activate her histamine symptoms. Then she also has high ammonia and high hydrogen sulfide, both of which can be toxic to the gut. And she also has an elevated estrobolome, which means that she's really struggling on clearing out that estrogen and could use some liver support as well. So let's get into the chat. What do you think? What would you recommend for this patient? Hey, Sharon, it's good to see you. And I love that recommendation, HU58. We got some total gut restoration, absolutely. What else? We have some other products. Changing in diet, adding in fibers. Yes, I love this. Looking for toxin exposures. Prebiotics, IgG 2000. Herbal antimicrobials, Ruby, great option. Wonderful job, everyone. All right, PyroGuard, that's one of my favorites. Okay, so let's get into this protocol. Now, of course, this is just something that I've thrown together, and it's just a kind of a protocol of sorts, but a guide. Of course, you can add things as you would like or take away. So let's talk about the first product that we are going to include. Many of you mentioned this already, the Total Gut Restoration. This features Megaspore Biotic, Mega Pre, and Mega Mucosa. So this series, the Total Gut Restoration Kit, it helps recondition the microbiome, reinforce those beneficial changes, and rebuild the mucosa. The first step is going to be reconditioning with Megaspore. We are going to focus on competitive exclusion. So we're going to introduce those spores to compete with the pathogens and crowd them out. It helps increase those keystone species and increase both alpha and beta diversity. Mega Pre. This is our precision prebiotic blend. It has pre-digested oligosaccharides, and this helps really affirm that new microbiome that you've introduced with Megaspore. It's going to give you higher and more stable microbial populations, and it's going to increase short-chain fatty acid production. And then during that 3rd month, we are going to rebuild the intestinal mucosa with Mega Mucosa. It can reduce mucosal and intestinal inflammation. It can modulate mucosal immune response and reduce the pathogen effect. So here's the overall schematic of the total gut restoration. And then the next product I would add, so many of you mentioned this, Mega IgG 2000. This is our binder. Now, I mentioned this after the total gut restoration, but you are welcome to start, and I actually would encourage you to start this before or after, or excuse me, before or during the total gut restoration kit. You can start it along with Megaspore. But Mega IgG is so important because it binds and eliminates toxins. It has a really powerful blend of immunoglobulins that can bind lipopolysaccharide. We talked about that earlier, the toxin that causes inflammation. It can bind C. diff toxins as well, along with H. pylori. And it is stronger than colostrum. Mega IgG 2000 contains 45% IgG immunoglobulins, whereas colostrum only has 25%. And unlike milk-derived immunoglobulins, it is free from bovine serum. So that means that it is lactose-free, casein-free, and beta-lactoglobulin-free. And of course, it neutralizes LPS, which circumvents the immune activation and helps reduce the inflammatory cytokine cascade. So we have the total gut restoration to balance the microbiome. That's going to address that gut dysbiosis that may be at the root of her hypothyroidism. Mega IgG 2000 is also going to bind up those toxins and pathogens as well. Now, let's add Mega Marine for some anti-inflammatory activity. Mega Marine is our gut-specific fish oil that is formulated with a superior ratio of EPA to EPA, DHA, and it also has the forgotten fatty acid DPA, all of which supports a healthy immune system and gut barrier function. Now, I included Mega Marine because it really does a beautiful job of balancing the arachidonic acid and EPA ratio. Now, this ratio is a biomarker of cellular inflammation. And high ratios of this fatty acid is associated with colon cancer, breast cancer, insulin resistance, depression, and heart disease. So we've got an anti-inflammatory effect. TGR is cleaning up the gut. IgG is binding up everything. But remember, this patient has IBS and depression. So it's important to make sure that we are addressing the gut-brain axis. And that's where ZenBiome Dual is going to come in. This is a dual strain probiotic that has Bifidobacterium longum 35624. Remember on that slide where we talked about all of the bacterial strains that make neurotransmitters? B longum 35624 makes GABA. So that's going to help support our mood. And it also features 1714, which is a psychobiotic. It's a Bifido longum 1714 strain that acts directly on the brain. Here's a study that we did this year on Bifido longum 35624 in combination with 1714. And we found that this combination significantly improved IBS symptoms. In this 8-week study with 40 women with IBS and mild to moderate anxiety and/or depression, we found that we were able to significantly improve all of these symptoms including depression scores and anxiety scores and sleep quality. And we even looked at inflammatory biomarkers like TNF alpha, interleukin 6, and C-reactive protein. What we found is that treatment or the supplementation with ZenBiome Dual led to a normalization of cortisol, our stress hormone. We also saw improvements in those inflammatory biomarkers, the TNF alpha, the CRP, and interleukin 6, which we talked about its impact on cortisol expression. So that is our discussion for the case study. In summary, the composition and diversity of our gut microbiome plays a significant role in the regulation of the endocrine system. Overabundance of inflammatory bacteria can upregulate inflammatory cascades and exacerbate endocrine dysregulation. When we choose to optimize the gut microbiome by increasing diversity, increasing short-chain fatty acid production, and supporting immune tolerance, we are then able to support healthy endocrine function in our patients. And I'm sure the team has already put our coupon code in the chat for you, but now you can write it down really quickly. The coupon code is here, and it is set to expire on November 3rd. So that gives you plenty of time to get some of the products that I mentioned today. And I appreciate you. Thank you for your attention. And now we can start getting to the question. All right. I'm glad to hear that, Ruby. That's awesome. And Sarah, oh, the code, let me see if I can find it. TS-DDW-OCT, like October, 23. That's the code.
I think I was talking with my mute on. We'll also send that in our follow-up email.
Awesome. All right, let's get to the questions. A question from Judy. On the hormone producers slide, I noticed tryptophan was not on the list. In addition to food sources, would tryptophan producers or production of metabolites for serotonin or melatonin acceleration also be markers to look for, especially with sleep? Great question, Judy. So yes, I do not have the list of tryptophan producers readily available in my head, but I will tell you that on BiomeFX, we do look for indole production in the microbiome. Now, indole production is very important because the gut and the body uses tryptophan to make serotonin, melatonin, and indole. And so by measuring indole, that's an indirect way for us to measure the ability to support tryptophan balance and tryptophan metabolism. I hope that answers your question. All right. What was that about kiwi? Oh, yeah. So on that slide, I was just saying that the factors that cause a negative change in the gut, like smoking and exercise, that can cause pathogens to overgrow, and then they can translocate across the intestinal barrier and cause inflammation. Or if you have kiwi, that's a good fiber that can grow healthy bacteria. And instead of making a negative impact on the gut, kiwi is able to make a beneficial effect on the gut and help tighten up, feed the good bacteria, and then they make short chain fatty acids that improve the mucosal layer or in the intestinal barrier. All right, question from Laura. What if beta glucuronidase is high? I think I mentioned that, but beta glucuronidase, when it's high, some of my favorite things to do, I give them calcium D-glucarate. That has been shown to reduce the expression of beta glucuronidase by 50%, but it doesn't just inhibit the production of beta glucuronidase in the gut, it's systemically. So it's not something that you would want your patient on long term. And then also liver support. So you can use your favorite liver support product. I'm a big fan of anything with sulforaphane and dandelion root and burdock root and all of those awesome liver supporting herbs. Okay, can you explain the beta glucuronidase and thyroid hormone connection again? Yes, so thyroid hormone can be detoxified a number of ways in the liver. Predominantly, it is detoxified as like a peptide, but sometimes the liver can use glucuronic acid as a way to detoxify the thyroid hormone. And so if you have a lot of beta glucuronidase, the same way estrogen would recycle back into the body, thyroid hormone can recycle back into the body as well. And so if you think of a patient, I mean, I've helped patients with thyroid conditions. And when you're first trying to find that dose of Armour or NP thyroid, it can be hard to figure out which grain dosage is going to be enough to kind of reduce their symptoms without throwing them into hyperthyroidism. And if you're finding that you're working with a patient that is having a difficulty finding that thyroid balance, then you want to look at the gut. I hope that helped. Please discuss the relationship between thyroid disorders, hypothyroidism, and H. pylori. You know what, Judy? Off the top of my head, I don't have a lot of details about H. pylori and thyroid disorders. My assumption is that, H. pylori does change the microbiome. It does change the gut and particularly the stomach and small intestine. And so perhaps the shift in inflammation shifts immune tolerance and affects the development of antibodies against thyroid. Vicky, I'm surprised the Lactobacillus species increases thyroid antibodies. Does this mean that this strain should be avoided in people struggling with thyroid problems? Now, remember, the Lactobacillus species that are associated with thyroid hormone antibody production, there are only a few of those Lactobacillus species. So not all Lactobacillus strains cause an increase in thyroid antibodies. It's just a few. And so that tells us, and also it's through molecular mimicry. So it's really just the immune system saying, oh, that looks like a pathogen. So let me just create an antibody against that. So what I would do is that I would be very careful, write down those two or three lactobacillus strains, and then just be very careful when choosing a lactobacillus-based probiotic, because I know that there are a lot of companies that put a ton of lactobacillus strains in their products. So just take a look at those. But if you have someone who is balanced with their thyroid, even if they have thyroid disease, it shouldn't be that much of a problem, especially if you've already improved their gut. Great question. Is intestinal alkaline phosphatase dependent on zinc? I believe zinc is one of the cofactors, and I also think B6 may be one as well, but I'll have to triple check that. Can thyroid medication be lowered or eliminated when microbial gut balance is achieved? Well, Melody, I think that depends on the severity of their thyroid disease, how long they've had the thyroid disease, and also, especially if it's autoimmune mediated, it's just depending on their ability to clear those antibodies and their ability to have a thyroid balance. So could it be lowered? It could. Could it be eliminated? I'm sure that's also possible, but really the goal is to make sure that they're stable throughout and maybe not necessarily eliminating medication. How are we on time?
We're just a little bit over, about
Okay.
11:07. So let's go for about 5 more minutes.
Sounds
And like we said, we'll be sure to send everything in a follow-up email. And I'm sure if you have questions, Sarah can also let you know how you can contact the clinical team.
exactly. All right. So next question. In regards to akkermansia, high levels are linked to metabolic syndrome. So I guess the balance is key when it comes to metabolic syndrome or type 2 diabetes and akkermansia. What are your thoughts on this? I share the same sentiments. At Microbiome Labs, our paradigm is believing in balance, not necessarily eliminating everything. And so that philosophy applies to pathogens, but also keystones. Yes, akkermansia is a mucin degrading bacteria. In healthy levels, it helps with the metabolism and growth of the mucosal layer, but if they are overabundant, they can cause damage. So it is all about balance, and that's where the keystones come in. So our keystones, if any of you have ever done a BiomeFX consult with me, you know that I describe our microbiome as a party, and our keystones are our microbiome bouncers. So they're going to welcome in lactobacillus and the spores, and bifido, come on in and enjoy yourself. But C. diff, you can't come in. Klebsiella, I don't think so. So the more keystones that you have, the healthier your microbiome is, and the more balance you can have in the microbiome. Do practitioners get a discount on BiomeFX? That is a good question. I know that if you were at, I believe if you were at MBKS, maybe there was a discount floating around. Actually, don't quote me, that might be incorrect. But I'm not sure on the discount for BiomeFX. Maybe the sales team can chime with that. Oh, you're still muted for some reason. Yeah. Did you press the button on the there we
Sorry,
go.
we're trying out a new microphone here, so I want to make sure everyone can hear me. If you can reach out to your sales rep and they can give you all the information on BioFX2, they might be able to. Again, don't quote us on that, but they can work with you on the BioFX prices. I know if you order a couple, you will get wholesale pricing, so there's always that option.
Nice. Awesome. All right, Cynthia would like to know, can it contribute to ovarian cysts? The estrobolome can contribute to ovarian cysts. Ovarian cysts tend to develop in an estrogen dominant environment. And so the estrobolome drives us into an estrogen dominant situation. So yes, having a high beta glucuronidase and an elevated estrobolome could possibly contribute to ovarian cysts. Question from Deborah, how does perimenopause or menopause affect the hormonal process that you're presenting here? Wonderful question. So a couple of slides in the very beginning, I talked about the neurotransmitters and how they have an impact on bacterial growth and biofilm growth and also virulence. So if you go on those slides, you'll see at the very bottom, estrogen or estradiol and estrone are shown to have an impact on virulence. And so with women who are perimenopausal and menopausal, you're losing that beneficial and protective effect of estrogen. Of course, your adipose tissue can make a little bit of estrogen, but it doesn't quite compare to what our ovaries were doing. So as you are improving the gut, you also want to try and support their hormones, whether it's through phytoestrogens from herbs, also Vitex, just other things to help support their hormones along with the gut. I'm so glad you asked this question. What is your favorite test for the gut microbiome? And that's BiomeFX. That is our favorite test. It is run by whole genome sequencing. So we look at the entire genome of the microbiome, and we look at the patient's genetic capability of making vitamins, digesting their food, producing different hormones and neurotransmitters like histamine and GABA. So that is our favorite test for the gut microbiome. All right, for estrogen recycling, if the recycling is high due to dysbiosis and more estrogen is reabsorbed, would you always see elevated estrogen on saliva or blood levels in order to be able to say that estrogen dominance is impacting symptoms? Or could they have related symptoms of estrogen dominance without actual hormone levels being high? Really great question. So you could have estrogen dominance and have normal estrogen levels, but low progesterone. So estrogen and progesterone, they exist together and in a balance. And so if you have low progesterone, but normal estrogen, that's going to put you in an estrogen dominant state and drive estrogen dominant conditions. But if you could also have high estrogen due to the reabsorption from the gut and other issues. So it can be either or, and that is where testing comes in. So you don't just want to test for estrogen, you test for estrogen and progesterone and a series of other hormones so you can get the full picture. Labs like the Dutch test is a perfect option for this. In Canada, we can't get IgG 2000 or megamucosa. Any adequate recommendations? Beth, off the top of my head, I would recommend any type of binder. Unfortunately, you're probably going to have to go with, and I say unfortunately because there are some scheduling things that you have to use with these products, but like something like activated charcoal or some other binder, but you will have to have them take that away from other supplements. With our spore products, do you need a binder? If they have excess pathogens or dysbiosis, yes. Yes. You do need a binder, and IgG 2000 would be perfect for that. I think we only have time for one more question. Let's see. Good question, Ruby. Is ZenBiome recommended after the TGR protocol? It could be after or during, just depending on what the patient is tolerating or going through. What we recommend is that you take the ZenBiome products that have bifido species, take them away from the spores so that the spores have time to make their effects on the gut, and then the bifido species has time to do that separately. And I think that's all the questions I could get to today. If you have more questions, feel free to reach out to your rep and they will put them in our question queue called Zendesk, and we'll be able to get those answers to you. I hope you enjoyed today's talk. I loved chatting with you all today. Thank you.